diabetic medication, and a particular outcome of interest, diabetic ketoacidosis. And we're going to walk through that example to try to give life to what these programs can do. And then we'll have some question and answer period. So essentially, what are we talking about in terms of a routine computer program, a routine query? So we start out with this idea that there's a template. There's a common kind of question that you might always want to know. For example, how many patients do I have that look like this, that are of this age range, that are exposed to this medication, that have had this kind of event? Those kinds of things are things that you would ask almost all the time, and they form the basis for the standardization of the computer program. So one example of that is just to say, what is the time that I am exposed to a product and I'm at risk for having experienced a particular outcome? So what we do with that is then we parameterize that program, meaning that we put input files in at the time of execution, and we run the computer program, and we do this repeatedly, and the idea behind it is that we can have more quality control that way. Because what we do is we pre-test and we validate that program with dummy inputs. And so then every time we add new input files, we can just rerun it, and it provides us with sort of a standard response every time. It also significantly shortens our turnaround time because all the computer programming is already done. Really what we're doing there is we're specifying the inputs to that computer program. And so we have the standard output that people become accustomed to. So folks may remember this from their childhood. These things still exist. The idea of Mad Libs, right? So this is the idea that we're getting at. We have a base computer program. That base is what you see here. So we're going to identify patients with a fill in the blank. So here we're looking for, is it a new dispensing or is it a prevalent dispensing of interest? And we're going to say, in order to count this individual in my cohort, they have to be of a certain age. They have to have a particular type of coverage. So that's something that Jeff and Kevin both talked about this morning. What we mean by that is insurance coverage, meaning do we have capture of that individual and their information over a particular period of time? And we're going to keep looking for that. And then there was also a woman who asked a question this morning about, well, how do we say what's a new user? Well, that's also a parameter that is in our fill in the blank, where we say, I'm going to define a new user as having not used this prior product in a certain number of days. So when we fill this in, now you can see that this is the parameterization space. So now an investigator or an epidemiologist has come in and said, this is how I want to utilize this program. I want to fill in these blocks in blue, saying I want to have a new dispensing and I want to have a particular kind of medication, in this case an SGLT2 inhibitor. And in order for me to actually continue to follow this patient, I want to be able to see that I've seen them for 6 months prior. I'm given this index dispensing, that they're an adult, they're not a child, they're between 18 and 100. And that they haven't received a prior dispensing of that same type of medication in a particular baseline period. And that's what I'm going to describe as them being a new user. So what new use in this case means is I haven't used a prior medication in that class or a related series of classes in a 6-month baseline period. And so we have a couple of different kinds of these computer programs. I'm going to discuss them now, ranging from the simplest to the most complex. So the first kind is just a basic count kind of query. What this is looking for is presence or absence of a particular code in the data set. This is sort of, you could think of as your go, no, grow criteria, your first stop shopping. Do I continue to actually pursue this question in this database or not? And so this is a very quick turnaround query for us. It takes about a week. So let's say that, yes, we see that we actually have the existence of particular codes of interest in the data set. Now we actually want to do some more cohort identification. We want to look at these patients. We want to describe them in a certain way. We want to narrow them down by certain criteria so that we can follow them for a particular period of time. And what we call this, or we think of this as our descriptive or our feasibility characteristics. And so we'll follow those folks, and we're going to look at things like how long have they been enrolled? So how long can I follow them for? Can I follow them for a year? Can I follow them for 3 years? We can look at things like, have they had particular events of interest? And how long does it take to get to that event? So these are the types of things that we would do in a descriptive or a feasibility query, which we'll talk through in our training example today. Sort of the next sort of step of complication is, say I do this descriptive or feasibility query and I decide I actually want to pursue a formal study design, trying to get an inference using an inferential query where the design of that is to produce an effect estimate. So that's something like a hazard ratio, a relative risk, an odds ratio. And there are a couple of different ways that we do that. We have a couple of different computer programs available to us. I'm not going to talk about those particularly today. We previewed in the morning that we're going to have another follow-on training that's going to occur coincident with the Sentinel public meeting, which usually happens in February. And we'll be going into the inferential queries at that point. So this is sort of part one to a two-part training where the inferential piece will be discussed at that next training in February. And then finally, if I produce an effect estimate and I say a treatment group, users of a particular medication, as compared to a comparator group, users of another medication, and their risk for a particular event, so in this case what we're talking about here is diabetic ketoacidosis in our training example, then I might want to follow up and say, am I really sure that that person actually had diabetic ketoacidosis? And I can do that in one of two ways. One is to look at a profile of that person's claims history. So what that's looking at is from the perspective of the insurance company who receives all those claims, do they have a pattern of events that is commensurate with what the outcome is that they are being experienced? The other available thing is a chart review. And so that's actually going to a vendor, getting a medical chart, looking at it and validating that in fact that patient experienced that outcome of interest. So you can think of this whole process as a sequence or an arc. It begins with the idea that I have a safety signal or a public health question of interest. And then it steps through all of these pieces in a sort of a logical order to get to getting an answer to my question. And then really validating that answer in those last steps. So you can look at that as a feasibility period, an inferential period, and then a follow-up period. And what we're going to focus on today is the feasibility period and how to ask those feasibility questions of interest. Can the Sentinel Distributed Database, with the data that it has in it, actually say something about your particular issue of interest? So let's go to our training example to give us something to walk through. So we start out and we are going to talk about a particular exposure of interest. It is a novel anti-diabetic medication. It was first approved first in class in 2013. It is approved for use with diet and exercise to lower blood sugar in adults with type 2 diabetes. Its mechanism of action is that it lowers blood sugar by causing the kidneys to remove sugar from the body through urine. Not approved for patients with type 1 diabetics. It's only a type 2 medication, and there is a listed example there of some of the particular brand and generic names for different medications in that class. So this is just a timeline to sort of give you a sense of what we're talking about. There were 3 new medications in this class approved since 2013. And so you can see the sequence of the availability of this medication to the general population. As the medication became available to folks, there started to come in case reports or something through the spontaneous reporting system. You can think of them also as passive reports. It's called the FDA Adverse Event Reporting System, or MedWatch. So these are issues where someone says, I took this drug and then I had this outcome and I think they might be linked. And then they write some information in this MedWatch form and send it into the FDA. And so enough of those had come in that the FDA decided to issue a drug safety communication in early 2015. And what they said in that drug safety communication was that SGLT2 inhibitors for diabetes may result in a serious condition of too much acid in the blood, which is another way of saying diabetic ketoacidosis. And so here's a screenshot of that drug safety communication. And so now this is sort of our motivating question of interest, our motivating exposure outcome pair, our safety issue that we want to think about. How can the Sentinel Distributed Database advance our knowledge of this particular situation? So we start out with, again, what was the issue that drove the safety signal? You have this, what we call case series in the FDA Adverse Event Reporting System. So there are 20 different cases of diabetic ketoacidosis among patients who were treated with SGLT2 inhibitors. This was a serious condition, so all of these patients in this case series required ER visits or hospitalization. Their median time to onset was fairly short after their new exposure. So you were looking at 2 weeks. So basically they start their new drug, and then if they're going to have this event within 2 weeks' time, they end up having this event. So one thing that was noted in the case series was that it wasn't really a typical presentation of diabetic ketoacidosis. So diabetic ketoacidosis, although I'm not a clinician, I'm married to one, so I pre-briefed this morning to make sure that I had all my facts straight. But diabetic ketoacidosis is, at least in part, caused by having insufficient insulin on board. And so oftentimes people are looking for a type 1 diabetic who's missed some of their insulin doses or has had a recent change in their insulin dosing. And so typically in a type 1 diabetic, that condition of diabetic ketoacidosis is often accompanied by a high glucose level. And together, this presentation in the emergency department causes somebody to clue in right away to, oh, this is a diabetic ketoacidosis. I need to get this person's insulin under control, because typically the treatment for that is to actually then provide insulin. And so what was unusual about these cases were that most of them were type 2 diabetics, and that the glucose levels they presented with at the time of diabetic ketoacidosis were only mildly elevated. And so as a result, there was this delayed recognition of the condition. And most of the physicians reported that they didn't expect it because these were folks who were using a new oral anti-diabetic medication that was intended for type 2 diabetics. And so the unusual presentation adds to the safety issue in this case. And so then we get to the question of how can Sentinel and the data in the Sentinel Distributed Database help to further generate information about this relationship? So we can ask two very simple feasibility questions just to start out. So the first question is, well, we have this question about SGLT2s. How are people using it? So are they getting one dispensing and then continuing to pick up further dispensings? Are they actually using it as a chronic medication? Or do they start it, then they stop it, then they try a different type of oral anti-diabetic medication? So we just want to understand a little bit more about how it's being used in real world and real life practice. And then another question that we could ask is, with respect to diabetic ketoacidosis, is are we seeing this more frequently after certain kinds of exposures? One of the drawbacks of the FDA adverse event reporting system is that there's no what we think of as a denominator. So we don't uniformly measure everyone and then see how many people actually had this event. We're taking in reports as they come in, and those may or may not reflect an entire population. So one of the advantages of having an observational database where you can look at everybody who meets certain criteria is you can measure all of them for whether or not they're having this outcome and whether it seems to be occurring more frequently following particular types of exposures. So let's go back to our training this morning. We're going to use our Stoplight Rubric again. So when we think about, can we at the very beginning even look at this in the Sentinel Distributed Database, we want to think about what type of an exposure do we have. And in this case, we have a self-administered outpatient pharmacy dispensing. And as a result, that's a green light, so we should go forward. We also want to think about our outcome, diabetic ketoacidosis. And so what we're looking at here is a medically attended outcome. In fact, this outcome was seen in the ED or the inpatient setting. It was hospitalized, so it was very serious. It had an acute onset, so in case the information we get from the case series is that it was happening within 2 weeks of exposure. It had clear diagnostic criteria. People are suffering from an imbalance in their insulin. And it's easily identifiable in claims data. And so this is also a green light. We continue with sort of this example and think, yes, the Sentinel Distributed Database has a good potential to be able to add information about this particular case. So now our challenge is, how do we make this public health question transform into a computer program to actually get us the information that we want to have? And so I'm going to go back to this slide and say, in the interest of time, we're going to breeze through that initial summary table piece. This is the counts piece where I just look in my data and I say, are there any counts of anyone who's been exposed to an SGLT2 inhibitor? We did do this for this query. I'm not going to talk about it here just because I want to sort of speed through to the next step. And the answer is we get back information that says, yes, in fact, we have people with evidence of this drug use during this particular period of time. So we're going to go right into that cohort identification and descriptive analysis query, our feasibility query. And so we are, again, thinking about two questions, both of which can be answered with the same computer program, the one that's designed to grab cohorts and describe information about them. And so we start out with our first question, which was asked earlier this morning, so thank you to whomever asked that, of how do we define a new user? And so whenever we're looking at new user cohorts, we're starting with the idea that we need to have a period of time where we look back, which we would call our look back or a washout period. And during that time, we're looking for evidence of a prior dispensing of that medication or a prior dispensing of a related medication, if that's the case. And we need to make sure we have enrollment during that time, so we have complete coverage of information about that person for that period of time. And so in this case, what we have is we're seeing an initial dispensing in this training example on January 31st of 2014. We're going to look back here 90 days in this particular example, and we're going to see that they didn't have evidence of another dispensing during that period of time. So for this purpose, we're going to call them a new user. Keep in mind that that 90 days is specified by the epidemiologist of concern, so it could be 90, could be 183, could be 365. It's however the person who's asking the question wants to specify it. So then we think about how we define a treatment episode. So we talked about this a little bit this morning. What we're doing here is we're taking claims data and we're trying to reconstruct a period of exposure for this person. And what do we have? We have a dispensing here, we have a dispensing there, we have a dispensing there, and it's our job to string together this band of dispensings and say this person's either exposed or they're unexposed to this drug during this particular time. And so in an idealized example, we have 30-day dispensings, which are typical. They basically get dispensed their 30-day dispensing, they use it perfectly, and then on the very day that it is done, they go and they pick up the next 30-day dispensings, and then again, they use it as described, and then they pick up their next one. Gotta tell you, in real life, this almost never happens. And so this is an example of, if you had sort of perfect people who did exactly as they were told all the time, this is what you would get. You get a 90-day active treatment episode. But what we do have is we have people who either pick up early or pick up late. And so what we first have to think about is what do we do with the people who pick up their drugs early? And they can pick them up for, of course, many reasons. They might be traveling, there might be, they just want to be ahead of the game. And so what we have, though, is the way it appears to us in the data is we have these overlapping periods of dispensing. You got a 30-day supply, but then I see you have evidence that on the 20th day, you picked up another dispensing. And so I could call that 83 days of treatment. I could shorten it. Or I could basically string these together and call it 90 days. And that's what I backed up. And so that's what we end up doing, is we do what we call stockpiling, which is that we line all the prescriptions up and we assume that people take those prescriptions, and because they're refilling on time or early, that they're actually utilizing them as intended and they're just ahead of the game. So the stockpiling algorithm adjusts all of those dates together to create a treatment episode to ensure that we have non-overlapping days in supply. And there are a number of specific technical options for that I'm not going to really get into here. We also have the converse. We have people who actually pick up their prescriptions late. So they have a 30-day dispensing, and then they get busy and they don't do something, and then 3 days later they're gapped in their medication, and then they go pick up their next dispensing and then start taking it. So then the question is, do you consider that gap to be a period of time where they're not exposed? Or do you think that treatment, that basically that biologically, that drug is still in their system and they're still at risk of having an event? And so you have to define what to do with those gaps. So here we have a situation where we have a person who's gapping and they have 3 days in between their dispensings or 5 days. And the question is, we allow the user to specify, do we say this person has been continuously exposed to this medication? Or do we say that we have no tolerance for gaps, and anytime you gap, we're going to stop considering you exposed? That's a choice that the investigator has, that the computer program makes possible, but it's something that we have to account for. Finally, and some things about the gaps, you can be fixed, or we can also do some more complicated gaps with respect to percentages. It also can be 0. Finally, there are times when people actually really cease treatment, but you want to account for them still being at risk for having an outcome of interest because we think that biologically that they are still exposed to that product. So basically the idea is that if you're taking a drug and on the 30th day, you don't pick up on the 31st day, let's say, you don't pick up your next dispensing, you still have that drug in your system. You still may be at risk of experiencing an adverse event. We have to decide when to count you for being at risk, and that's what we think of as our exposure extension. And so we add a piece on to the end of the last dispensing and say we still think you might be at risk for experiencing this outcome of interest at that point in time. All of these pieces together, the actual dispensings, the gaps, and the extensions, you compile them all, and that is what we call a treatment episode. So this is a period of time when a person we consider to be continuously exposed to a product of interest. So when we have a treatment episode, we might decide that for our particular cohort, we want to keep either people in or take them out based on what's happened to them in their baseline period. And so for cohorts, we can also put in inclusions and exclusions. And what those do is they basically say for some period of time prior to that new dispensing, I'm going to either kick you out of my cohort or keep you in my cohort. So let's go to our SGLT2 example. One of the things that we could do is we could say, I want to look at any new starter of an SGLT2 medication. But then I might want to do a sensitivity analysis that says, I want to kick out in my cohort anybody who has a type 1 diabetic code in that period of time. Or I want to kick out anybody that has insulin as a prescription during that period of time. So you can set all of those things as you go through as either a primary analysis or secondary analyses in order to basically more specifically narrow your cohort to exactly what you're looking for to that period. The only part of that that's really important is you have to have enrollment during that period to assess the presence or an absence of a code to actually define your cohort. So we'll talk a little bit about the query specifications here. So again, this is the inputs, the input files or the input parameters to your computer program. And so in this case, in our example, we're going to be looking at SGLT2 users. So right now I have two different scenarios, one that separates out canagliflozin from dapagliflozin, I hope I said that right, with medical and drug coverage that was required for 183 days prior to their new dispensing. So essentially, I'm defining here my new user as having not been on an SGLT2 inhibitor for the 183 days preceding their incident dispensing, which I'm going to call their index date. And I'm going to use a standard stockpiling algorithm, which means I'm going to allow people to pick up early and I'm going to string their episodes together without having any overlap in days. And I'm going to allow people to have a gap of 10 days between picking up their next dispensings. And when their last dispensing, when I have evidence that they have a last dispensing, I'm going to still kick on 10 days at the end. And I'm going to call that person exposed during that part of time. And so now what's going to happen is I have my computer program. I have it parameterized in the way that I want. At the Sentinel Operations Center, what we're going to do is we're going to have a second check of that parameterization to ensure that we think it's doing what the requester asks. Then what we're going to do is we're going to send those computer programs out to our data partners like Kevin and Cheryl over here and some of our data partners that are in the audience. And they're going to receive those computer programs and they're going to execute them behind their firewall. And then they're going to produce results. They're going to review those results. And then they're going to send them back to the Sentinel Operations Center. We're going to aggregate that information among the participating data partners who return our query. And then we're going to provide that information as our output. And this is the output that we're going to show for this question. So let's remember back to the question. We were thinking about just what is the general pattern of use for these particular products? What can we say about them? So we have a pretty sort of wide-ranging query, meaning that it's sensitive. We're not kicking a lot of people out. We just want to see anybody who we consider a new user, what's happening with them. So the first thing we might want to think about is how are we doing in terms of uptake? Are people actually using this new medication? And so in this case, you see these flat lines here for cumulative new users. This was an indication of when this query was run, which is actually quite some time ago because we use it for training. And so this isn't really accurate to what data we have now. This was run a bit ago. But that flat line means it's sort of end of data, which is when the data stopped at the data partners. But what you can see for the two drugs, canagliflozin is the first in class, dapagliflozin is the second in class. And the first mover in this case of this drug had a bigger uptake, which you might expect because it's the first, it's the novel one, it's the one people start on, it was available earlier. And so it has a bigger uptake. Another thing you might want to ask is, again, what is the pattern of people who are on this drug? So what this is a survival curve. It looks a little complicated, but really it's not. Along the x-axis at the bottom are the total number of days in our treatment episode. Remember, we're creating our treatment episode by stringing together this criteria and saying we're exposed to this drug. And what you can see here is a big drop at day 40. So what's happening there? Day 40 in this case happens to be a first prescription of a drug, which is usually a 30-day prescription. And then I had that 10-day exposure extension I tacked on to the end to say that I think that a person who's been on this drug for 30 days still has it in their system for 10 days after they've actually stopped taking a pill. And then I don't follow them anymore. So what does that mean? I look at this and I say, you know, of new users of canagliflozin, I'm at a 50% survival, meaning half of my cohort has not picked up their second prescription. And for the dapagliflozin, it's even worse. 40% of my cohort actually goes to pick up their second prescription. Now, I don't know exactly what this indicates in terms of, what people are doing or why they're doing this or whatever, but what it tells me is if I want to look at a particular exposure and outcome pair of interest, I better hope it happens really close into when they pick up that initial dispensing, because I'm going to lose information on them as soon as I start looking for that second dispensing. And so it helps me to understand whether the data themselves are capable of answering my question of interest. So then I want to look at information about their drug dispensing patterns. So one thing that's true about both of these drugs is that they're typically given in a 30-day supply. And there is some portion of them that are given in a 90-day supply. And actually, you might not be able to see it. There's little dots there at the very bottom for these in-between values, but it's almost nothing. It's dwarfed by the 30-day supply or the 90-day supply. And so what are my take-homes from this feasibility query? So let's remember that in the FAERS data, diabetic ketoacidosis occurred with a median time to onset of 2 weeks, which means that even when I'm looking only at a singular 30-day dispensing of these new types of oral anti-diabetic medications, I can still capture that DKA event in that time period. Now, let's say the event of interest of mine occurred 3 months or a year after the initial exposure. I'm kind of out of luck in these data. I need to go find another data source because these data aren't going to do it for me. Some things that I could do is I could increase my extension parameter or my gap parameter. I could do some sensitivities to see whether I'm going to pick up more capture at that point. But it's really important to understand that then you could potentially be changing the clinical meaning. One of the things that we do in creating these computer programs is we try to take out all of this, what I would call the scut work. We basically try to standardize all of the things that people want to do every single time, but what we're not trying to do is take away the thinking. And so it's really important to still think about all these parameters and how you put them together to get to your conclusions. And that's the reason why some of these parameters and how you set them are really important. So the next question, because we already talked a little bit about the general pattern of use, is to look and see if the outcome is occurring more frequently after particular kinds of exposures. And so I'm going to look at a set of query specifications again. These are the input files or the input parameters to my computer program. So I have 3 different classes of oral anti-diabetic medications up here because I'm going to be looking for differences in class, saying how is diabetic ketoacidosis occurring after SGLT2 inhibitors versus sulfonylureas versus DPP4s. And I'm going to again require that I have medical and drug coverage for the 183 days in the baseline period to establish that someone is a new user. In this case, I'm still going to use that standard stockpiling algorithm, and I'm also going to use the 10-day gap that I had before and the 10-day extension. Here I don't have it listed, but in this particular query, we also required people to be incident to the other classes, meaning that if I was going to be identified as a new user of an SGLT2 inhibitor, in that 183-day baseline period, I could have also no other dispensing of sulfonylureas or DPP4s. So I was looking at people who were having sort of a monotherapy situation where they were choosing an anti-diabetic medication and they weren't on several others at the same time. The only exception to that was that metformin was allowed for all of the users at any time. And so now, again, at the query specification stage, what I'm doing is I've got my computer program, I've parameterized it, I'm going to have someone second check that information. Again, as a Sentinel Operations Center, we're going to send that out to our data partners. They're going to execute that program on their data sources. They're going to produce results. They're going to send that back to us at the Operations Center. We're going to aggregate that data, and then we're going to provide information on that aggregated database as our output. And so what we're going to look at here on this next slide is information about the rate of this outcome occurring after new use of each of these 3 types of oral anti-diabetic medications. And so here's just a display of my cohort criteria. So again, I'm going to have no use of any of these study medications in these 183 days. I'm going to have a point that I'm going to call my index state where I have my incident dispensing. I'm not going to actually have any criteria that's going to limit my cohort with respect to type 1 or type 2 diabetes. I could. I'm choosing not to in this point because I'm looking for a more broad spectrum group. Then I'm going to look for an event. So once I have my initial dispensing, now I have to think about, okay, how am I going to find my diabetic ketoacidosis event? And so then there are some things that we have to talk about with respect to parameters that take care of the event. And so here the event is going to have a period of time in between my dispensing and my event, which I'm going to call my time to event or my time at risk. It's going to also be my follow-up time. I'm going to say that I want to see people who have a new experience of diabetic ketoacidosis, meaning that I'm not going to look for people who seem to be at higher risk or who've had prior in a particular period of time. So I'm not going to look for people who 1 month prior to their new oral anti-diabetic medication had diabetic ketoacidosis and then experiences it again. I'm going to require people to have a washout and to say, I'm looking for a new onset event. And I'm going to describe that new onset event as being the first in at least 183 days prior to the start of my medication. Also with the event, I have to say, where do I want to find it? So again, I'm looking at claims data and I have several choices. I can look for it in an ambulatory setting, I can look for it in the emergency department, or I can require that somebody be hospitalized. If they're hospitalized, I can require that it has to be the principal discharge diagnosis, the reason that they were in the hospital. And so all of those things are parameters available to me that can help me better specify the question that I want to ask. And so here we're going to look at a set of specifications again. We're going to say this is diabetic ketoacidosis. This event code is an ICD-9 era event code. Again, we ran this training query and we prepared these training slides quite a while ago. We haven't yet incorporated ICD-10 era, but now we would. If we were going to run this query today, we wouldn't run it looking at only ICD-9 era codes. We would look at ICD-10s as well. That event and incidence criteria relates to saying, I'm not going to have had that event in the prior 183 days. In this case, I'm going to look for any evidence of that event, meaning in all settings prior, any event that happened is going to disqualify me from being able to experience that event in the future. Additionally, when I look for the event, I'm going to look for it in the inpatient setting only. Why is that important? Because the FAERS case series that came in was talking about people with hospitalized DKA. So if I'm going to be keeping with that sort of safety issue of concern, I'm going to limit the DKA events I have here to hospitalized DKA, although I am not going to require it as my principal discharge diagnosis. So essentially, all of these, again, are parameters that are available to you to ask these questions and to help to better specify the cohort of interest. So here are some results. So it's a lot of numbers to look at initially. So if we start over on the left, we have our 3 different classes of oral anti-diabetic medications, and we have new users. The first thing to look at is just to say, SGLT2 inhibitors are the least new use of all of the ones there. But that sort of makes sense because they're new on the market and doctors may be hesitant to prescribe them at first. They may prescribe them to very specific people. And these other drugs have been around a lot longer and in some cases are a lot less expensive. And so it makes sense that we would have some of these other drugs that are more widely utilized during this period of time. The number of new users follows is a similar sort of order of magnitude as the total dispensings and the days supply and the years at risk on exposure. So all of these are numbers that help me to understand how much follow-up time I have, how long I can look at these people. I also have information there about new episodes that are followed by my event of interest. You can see there the days supplied per dispensing. So we already talked about our first query, looked at SGLT2 inhibitors and told us most people were getting a 30-day supply, with a very few getting a 90. So on average, this looks very consistent with the information I had earlier. You look at sulfonylureas and it looks like, oh, maybe there are more folks who are getting 90 days there. So we're stretching sort of our mean, getting closer to 90 instead of hanging out towards 30. It also may indicate something about the expensiveness of the medication because typically more expensive medications are dispensed in these 30-day supplies and they're not given for longer periods of time. And so then the sort of the big ticket information here though is what's my event rate? So how many of my episodes have events? What are my number of events per 10,000 person years at risk? So that's for the person time that I follow these people. What is this? And so what this can tell us here is, again, recall some of the limitations of the FDA Adverse Event Reporting System, or FAERS, which is that you don't have that longitudinal observational data that allows you to get at a denominator. And so now we have a denominator here. So we have crude, so unadjusted, class-based background rates that suggest that the FAERS signal can't be sort of explained away and say, oh, it's stimulated reporting. There's a brand new medication on the market and people are filing event reports about it because it's brand new. So we don't have evidence that supports that idea. We can say that, you know what, there's evidence here that says maybe further study is really required. And what we would do is we'd think about basically more extensive confounding control. Because right now, this is a crude rate that really is unadjusted. And so we have to take into account the fact that we have observational data. We have to do a lot in terms of confounding control to sort of wrangle that observational data into an appropriate effect estimate. And so we would want to do more careful study. One of the things that we would consider here is that right now in the Sentinel Distributed Database, as Jeff was speaking about this morning, we don't have the Sentinel CMS data in there yet. We're getting there. But right now, and at the time this query was run, we didn't have the CMS data in here. So what we have is an undercapture of folks who are over 65, who are often folks who are diabetic and would be trying out some of these new medications. And so when we think about what we're doing here, we're thinking about how well are we powered to actually do a formal study to get to a hazard ratio, to get to an odds ratio. And so that's part of the information here, is that we're using this for sample size calculations to decide whether or not to proceed with looking at this study in the Sentinel Distributed Database. And just a reminder here that of these rates, these are crude. They also did not account for people who are either only type 1 or type 2 diabetics. So this is all comers. Any person who took one of these drugs is included in this rate. So one of the things that's true is that in the initial look at DKA, we had a very simple definition. So we are looking at an any position, inpatient, ICD-9-CM diagnosis that was 250.1. That asterisk is a wild card. It means any number after that can be utilized. And so this is what we would think of as a simple list of codes with ORs connecting them in terms of Boolean logic. But what we might want to do is we might want to refine that definition by adding more criteria to it. What that's going to do is actually, it's likely going to increase your positive predictive value, because now what you're going to look for is you're going to look for a code plus certain other criteria. But what that's also going to do, it's going to reduce your sensitivity. So you have trade-offs there when you're thinking about how to define your outcome. And so one of the identification problems with this initial situation was that the clinicians, according to the FAERS reports, didn't initially recognize that this was DKA, because they were looking for DKA in the presence of hyperglycemia, because that was the usual presentation that they were used to. And so maybe what we want to do is we want to account for that in our outcome definition. And we could do that by actually saying that we have to have this type of code, and we can say that we don't want to have another type of code. We can use complex logic is the point, is that we can have condition A and condition B, or we can say condition A and not condition B. And all of this is available to us with the routine computer program that we have. This is just an example of an attrition table, so you can start to see that where you're losing people. So you start out and you say, I want to find anybody in my database that has data during this period. And then I'm going to say, I only want the people who have both medical and drug coverage. And then I'm going to say, I only want the people who have evidence of this exposure. And then I'm going to say, I only want to have people who have enrollment for a particular period prior to this exposure. And then we're going to further look to say that they haven't had that DKA event. And so the point is that this is the way, you know, in any typical cohort criteria plan, you're narrowing down your patient population. And why is this important? Because this gets to your sample size, your ability to go on and do further study, is how many criteria you're going to require to actually look at these folks. So today what we've talked about is feasibility queries. So what are these? Basically, this is our main engine, if you would, our computer program for cohort identification and descriptive analysis. It's a type of program that supports complex algorithm definitions. So what that means is that we can use and or Boolean logic in multiple settings in order to identify either an exposure or an outcome of interest. It stratifies that cohort automatically by age group, by sex, by site, that means the database itself, and by the month year of exposure. It automatically will generate a cohort attrition table similar to the one I just showed you. It also will generate baseline covariate characteristics. So one of the features that it has but was not run at the time of this query is you could just report descriptively, I want to know, prior to that SGLT2 inhibitor incident dispensing, how many people had a type 1 diabetes code in the 183 days prior. Now I can do that in what we call a baseline covariate characteristics table without actually requiring it of my cohort. I'm just really reporting on it. It's very descriptive in nature. It's the prevalence of that particular code. And I might also report on other characteristics of interest to me. The amount of heart disease that this particular cohort has, whether they have hypertension, whether they're on other types of oral anti-diabetic medications, whether they're on insulin, Essentially, I can report any presence or absence of any medical condition for which there is a code or any exposure of interest for which there is a code. And so I can grab all kinds of information about this particular group of people that can help me to figure out whether I can ultimately answer the safety question of interest or the public health question of interest that started this whole process. So I'm using that for sample size calculations. And so in terms of our response time, really what we really want to concentrate on is really thinking through what we think of as those specifications, those input files, really asking the right question and ensuring that you understand enough about your data, and we reach out to Kevin and Cheryl and other data partners of interest when we have questions about how these outcomes are identified. How is this happening in your data set? What does this look like here? We spend most of the time there, and then we have some time for implementation to go back and forth. So again, if we go back to our questions of interest, the idea here was to set up and say, I need to ask some baseline set of questions that help me to think about whether the Sentinel Distributed Database is well suited to actually advance the knowledge base here. Because really what we're thinking about is, do I continue on in this system, or do I go to another system which has different types of data that might be better suited to my question? And so after that feasibility period, the question is whether or not we proceed to that inferential step. And that's that third box there with the inferential design. Those codes there, that L2, L3, those are our codes for when we say we're doing a level 2 analysis or a level 3. It's just indicating a higher degree of complication. Because now what we're talking about is a comparative assessment, where I'm going to say, I have my treatment group of interest, SGLT2 inhibitors. I'm going to have to identify who I'm going to compare them to, which of those other classes of medications, or maybe basically another class that I didn't even have on this list. Maybe it's just metformin users. It could be whatever the user specifies. And then to actually execute that inference, the idea here being, again, more sophisticated confounding control. Because bottom line, this is observational data. All we produced in terms of the rates that we saw were crude and unadjusted. And so the role of inferential queries there is to generate effect estimates. And we have a couple of designs for that. Again, I'm going to touch on these here, but really the next time we'll talk about them is in February when we come back after the Sentinel annual meeting and go through some of these inferential queries there. So we have a propensity score match design, we have a propensity score stratified design, and we have a self-controlled risk interval design. And those typically take longer to specify. They're more complex queries and longer to actually turn around that information. If we're doing something that we call level 3, what that means is it's a sequential look. So a level 2 in terms of inference is a retrospective. So basically what we're doing is we're saying we're waiting for all the data to get in our database, and then we're looking back and we're doing an end of study analysis. A level 3 is actually where we're waiting for data to accrue. And that has multiple hypothesis testing that then we control for a total type 1 error over a period of time. But because we're waiting for the data to accrue, the time when you actually get to your effect estimate may be months or years from when you actually initiate your query. The idea here is truly to look from the time a drug goes on the market and say, okay, I'm going to check it out in 6 months. And then 6 months later, I'm going to look again. And then 6 months later, I'm going to look again. And so what did we talk about during this first session? We showed an approach to using our routine querying tools to help advance the knowledge base on a particular safety question of interest. We used a particular example, the idea being to try to help to get this ability to wrap our mind around this question and how we might utilize these data appropriately. And really what we use this for is to think about how early exploratory work is really important prior to going on to a more sophisticated analysis. And so we really recommend that we always do these sort of preliminary steps before we proceed further. And so we have time now, I still think, remaining before we have a break. And then we're going to talk about a new training example and medications during pregnancy after the break. So why don't we take now, since we have a few more minutes, so we're not out of time at least, according to our timekeepers, and take any questions from the audience. Yes, ma'am. Right, so some of those stratifications, like men and women, that's built in, so that's going to automatically pull that out for you, and others of them are not. Others of them are specified. So what we do when we get a public health question in or a safety question from, usually at this point, the regulator or another asker, is we sit down with them and we think about what are their primary analysis, what is the primary question of interest, and then we think about what are the sensitivities that you want to think about, and we call each of those particular runs of the program a scenario. And so we think about what is the total set of scenarios that you are interested in looking at, what variations are you interested in looking at that change relative to your primary analysis. And we try to narrow down to the variations that we think are likely to be of most import. And a lot of that can come in that feasibility stage, because if you bring back a bunch of baseline covariate characteristics about your cohort, you'll know how many people are type 1 and type 2 diabetics. You'll know a lot more about the population you're trying to measure. And so essentially, the more that you do in that prelim stage, the easier it makes it for you later when you try to set up to do an inferential query, because now you've gotten all this background information on those folks, you've gathered all that data, and you can make sense of it to try to go forward. Oh, I'm sorry, I forgot to repeat your question. I'll do it on the next one. Sure. So the question was, what does the turnaround time actually encompass? What are the steps to doing this? So what we think of as the steps are, the first piece is the specification phase, which actually we don't have on the timeline. That's the only thing that we don't time, because essentially that's the back and forth with the asker about what they're trying to accomplish, and having them write out sort of what you would think of as a protocol or a safety analysis plan, which doesn't have to be complicated, but it has to have some very specific pieces in there. How do you want to define your exposure? How do you want to define your outcome? How do you want to define your covariates? And so that back and forth period of time is actually something that we don't show as timed here. Once those specifications get finalized, what we actually do is we test that query, meaning that we test the parameterization with the computer program itself in our test database. When we test that, what we're looking for in the results is, did anything very bizarre happen? Was there an unintended specification that created an unintended result? And so what we do is we share that with our actual requester and say, here's the test data. Are these what you expect? So for example, these are sort of logic checks. Is this being utilized by an older population? Is this being utilized more by women or men? So things that will show up there that sort of are logic checks to ensure that we've actually, that sometimes even when somebody tries to specify what they want, they still end up getting an answer that isn't congruent with what they think they asked. And so we have that back and forth that's necessary. That's usually at least 2 weeks' time. Then we settle on what we think of as our final set of specifications. At that point, that's when we send it out to our data partners, like Kevin, like Cheryl. And then they have a turnaround time that's contractually obligated with respect to how long they can hang on to the query and time it in terms of when they turn it around. We try not to send them everything at once. So we try not to send them 10 queries on one day and split it up, and so that we send them, sort of meter it out as best we can, so that they can run the query on their system, and then they can investigate the results and respond. There are certain times when they're going to look at those results and say, I'm going to have to use some more of my time to clear these results, because there might be a sensitive question that's being asked, or there might be patient level information that's being requested in a very small amount. So there are things that will change that timeline there. Then they'll send that information to us, and then we spend about a week doing the aggregation and doing the reporting out. And then the reporting out is essentially creating an overview of sort of, this is what you asked, these are the specifications you have, here is your output, and that sort of thing. So that's sort of where the timeline gets broken up. So you could think of it as specification, testing, implementation, sending it back and forth, and then reporting. Sure, absolutely, and that's totally true, that there's an increase in time, because not only that, it's the testing, right? So when we look at the test data, if I'm going to test 40 scenarios, then I got to look at 40 results and look and see if those all make sense. There are other things that add to that timeline. If I have a complex outcome, so I want to test logic like I have condition A, and I have condition B, and I have condition C. What I actually have to do is we look for the data in our data sets, but if it does not there, we actually create it. So we create a situation where condition A exists, but B and C don't. And then we check to ensure that our program didn't inappropriately pick that up. And then we look for whether condition A and B occurred, but C didn't occur. And so essentially, the more that you add on to the complexity, the longer you're going to turn around that time with respect to, or you're going to add time to your, not only your testing phase, but then also your runtime at the data partners themselves and what then comes back. I'm sorry, I forgot to repeat the question again twice. So the question there was, did it add to the time, if I add more scenarios, does it add to the time that it takes to actually turn around the question? Go ahead, sir. So the question was, is there a difference in the turnaround time with the FDA versus other interested parties? So Jeff often says it, and it's true. We are nothing without our data partners. And so everything that we ask in terms of turnaround time and speed comes with the negotiation of how fast they might be able to turn around that information, how fast that's possible for them. And so there are times, we have sort of set limits about the times that we ask them to return queries to us. But we also have options which, we don't actually have a way of calling it, but I think of it as like calling DEFCON 5 or calling urgent, where if the FDA decides that there is a public health emergency, they can like flash a blinking light and say, we need this faster, we need this faster. And we communicate that to our data partners and say, this is something we need faster. And they actually are so responsive to us to actually get it turned around in that time. So there are occasions where something can run literally in a week or less, but usually that's because somebody's elevated that question of interest to such a point that it does turn around that fast. In terms of the turnaround time, though, the steps are the same. Whether the requester is the FDA or the requester is another interested party, we don't change the way that we do the specifications, the testing, the implementation, and the reporting. So the amount of time it takes should be very congruent, regardless of the asker. I hope that answers the question. So the question is whether the public or any user has access to the distributed database, and whether the gains that could be had from allowing other people to have access could reduce the amount of time or maybe increase efficiencies. Currently, the only sort of door that opens for the Sentinel Distributed Database is the door that comes from the FDA. We have other types of uses of Sentinel. So people who independently contract with us to utilize the data that's in the distributed database, but it's not through the Sentinel mechanism. So it comes through other organizations that contract with Harvard Pilgrim as a coordinating center to operate the same sort of system. And there is also a chance, so those organizations include public-facing ones like the NIH, and also private organizations that actually come in to ask us to do that. So if you're a part of any of those organizations, you essentially have access to a door. And so right now, I personally don't know of any, and I can look to Rich or Jeff or Bob, who's joined us in the background, whether there is any intent for there to be a completely open or completely public use of the database. I know of none at this point in time. Go ahead. So the question is whether there's a more broad spectrum look at outcomes of interest. And so typically the way that these questions come in, they come in from an FDA asker, and they're asking about a particular exposure outcome pairing of interest. And if they want to broaden those outcomes, then they tell us that they'd like to broaden those outcomes. But if they don't tell us to do it, then we don't have actually license or ability to go ahead and just look at them. There are a couple of other types of programs that we have that were not described today. We hinted at some of them this morning. Signal detection or tree scan, where we look at just any outcome in this sort of set of 6,000 and anything that pops up. But it's sort of a different idea there, because this is really talking about trying to answer a particular exposure outcome pair of interest where you've already pre-specified what that pair is. And that is a more searching or signal detection type framework where you're really only specifying the exposure of interest and you're looking for all comers in terms of the outcome. So there's a hand over here, I think, before. So the question is, how do we vet the analytic output of the data partners? So what we actually do is, and one of the things we didn't spend a lot of time talking about is we spend a lot of time in our quality assurance curating the distributed database that they send to us. So we didn't talk as much about this process, but we talked about it sort of briefly in passing this morning. Each data partner creates a version of the distributed database, which they think of as their refresh or their ETL. It's where they've transformed their source data into the structure that is required by the Sentinel Common Data Model. And when they transform that structure, we send a computer program to read that structure. And we look for many things with respect to whether they're compliant with the database. We have sort of a whole presentation on that. But essentially, there are over 1,600 quality assurance checks that run against that data. And the idea there is that you're vetting at that point the actual underlying data system. And so if there are inconsistencies there, if there are problems there, that we would identify them there, and we would ask the data partners to actually recreate that data prior to actually allowing it to be queried for a regulatory question of interest or a safety health question of interest. And so almost all of the curation and the vetting comes in at the front end on the data itself, which is actually one of the reasons why we can do all of the rapid querying, because once we've done that vetting, we've looked at all these data. We've looked at the patterns of the data over time as they continue to add more data and basically completely refresh their system to ensure that we think it's of the highest quality and able to answer regulatory questions of interest. Mm-hmm. Sure. So the tree scan program is one where we're doing still, I would call it methods work on both the drug side and the biologic side. So with the CEDAR and with CBER in terms of the centers. There are protocols that are available on the website with respect to future studies that are going to be using tree scan. And essentially, I think that we're at the point now where we feel fairly comfortable that it does what we intend it to do, which is that we have a particular design, a study design that we utilize most frequently. We're expanding into other types of study designs, and that you will see if you go check out the website of the different methods projects there. And we are working to continue to expand that design set, but essentially the idea is the same, which is you set an exposure of interest, and you're going to look very broadly at an entire range of outcomes, and you're looking for more frequently than expected occurrences of those outcomes following that exposure, and any of the points that merits a statistical signal or an excess of what's expected, which is pre-specified with a cutoff decision rule, that comes up for further investigation. So if something sort of hits that level, then it's then considered something that we think about further investigating through another mechanism. So. It has been tested and applied, and I guess I'm limited, and I have 0 minutes. I'm limited by what I can say to what's on the public website, but I would basically refer to what's available there, because that's what we have, that's sort of the latest we can share with the public. So folks, we're going to have a brief break, and then we're going to come back and we're going to talk about medication exposures during pregnancy, and we'll still have another Q&A session at the end of that where we can reach back to questions here if you still have them remaining. Thank you. Hi, I'm Judy.